All right, so in today's video, we're going to talk a little bit about the evolution of a population. And in this case, we're not talking about changes in creating new species, for example. I'm talking about slight changes in an individual trait within a population over the course of time. Okay, so in order to understand that, we have to understand the difference between what microevolution means versus what macroevolution means. So when you hear the word macro, like in macroeconomics, macro means kind of the big picture. When we're talking about macroevolution, which we're not going to go over in this specific video, but when we talk about macroevolution, that means large changes in species, creating new phyla, whole groups of different species. And that takes millions and millions of years. When we're talking about microevolution, though, micro means small. It's looking at small-scale changes within individual populations. We're not talking about speciation events here, really. We're talking about just a change in a particular trait over the course of one to several generations. Okay. So before we get into that, we have to talk a little bit about alleles again. Now, we talked about alleles when we went over Mendel. Remember, they're different versions of a particular gene, and thus cause slight variance in traits, or variations in traits. So how do we get these alleles? These alleles come about by mutations, random mutations that just happen to have an effect on that protein. Now, most of the time, mutations cause negative effects, like it causes the protein not to work better, it causes it to work worse. That's in most cases. So like when we're talking about blue eyes versus brown eyes, remember the brown eye allele is a pigmented allele. It causes pigment to be produced, where the blue-eyed one is when pigment is not produced. So it kind of the mutation caused that protein to not function anymore the way it should. But these alleles that we talk about in most instances or these new mutations that occur, don't just happen in your gametes that often. They happen in your skin cells, your nerve cells, your digestive, but those don't matter when it comes to evolution. These new alleles have to come about specifically in gametes. So this is only talking about new mutations that occur in gametes, and that's how we get new alleles. Remember, too, a quick review of mutations. There are the point mutations, which are the small-scale mutations where we change like one when you're looking at one gene, when you delete an individual base or a couple bases, or we insert a couple bases, or we just substitute one base for another, which can have varying effects on that specific protein that's produced by that gene, right? But we can also have large-scale chromosomal mutations where huge chunks of chromosomes or DNA are duplicated, translocated, deleted, inverted, and so on and so forth. So most variation in a population actually doesn't come from mutation. Now, all of those alleles that we're talking about were created initially by a random mutation, but how that allele spreads throughout the population happens through a couple different mechanisms. One is through genetic recombination, which is the main one. And we talked about a few different ways that can happen. Crossing over during meiosis, okay? Independent assortment during gamete formation, and then finally, the fertilization event itself, which is the fusion of two different individuals' DNA together to create a new individual, or two haploid gametes that fuse to create a diploid zygote that is completely genetically unique from either of those individual parents, but is a mix of those two. Okay, so now we're going to get into population genetics specifically, and what microevolution usually focuses around. It's the studies of changes in allele frequencies in a population over a given amount of time. So what does that mean? Okay, so let's say you have 10 wolves, okay? 10 of them or 9 of them are brown, 1 of them is white. And all of a sudden, we have a really nasty winter. So the frequency in this case, let's say the recessive trait is the white allele. So they would be like little b, little b. And the other wolves would be variants of that, big B, little b, or big B, because they are brown and that's the dominant trait. Well, if they have a really nasty winter and it's really snowy, that white individual is going to have a selective advantage over the brown ones because it is more camouflaged, and because it's a hunter, it's going to be able to get closer to prey items before they can escape, and therefore he will get more food. and he will have more energy, and he will survive more often, be more likely to mate than those other individuals. So over the course of several generations, we create more and more of these white, or the frequency of the white allele increases, specifically if that environment continues that way, like we have really nasty winters continuously. So what that does, it changes the frequency of that little B allele in that population over time. Before we get into the specifics of this and the math that we're going to talk about behind it, you have to know what the definition of a population is. So think about it. Think about what you would think the definition of a population is first. Okay. So the way it's defined, it's very similar to a species, though that's not a perfect definition, is that it's a group of individuals of the same species, so they have to be the same species, that can potentially breed with each other and produce fertile, viable offspring. That's the definition of a population. The next definition you have to know is a gene pool. If I were to, for some reason, get a pool, you can be sure that I would put that at the bottom of it just because that's how I am. Kind of weird. But what is a gene pool really? A gene pool is all of the alleles of all the genes of every individual in a population. Now that sounds crazy, but let's just pretend there's a population of 3 individuals, okay? One of them's big B, one of them's big B, little b, and heterozygous, and the other one is homozygous recessive, little b, little b. Well, then what we'll do is we'll look at the frequency of all the alleles total. So in 3 people, how many different alleles are there total? There are 6, okay? How many big B's are there? Well, the big B individual had 2, and the big B, little b individual had 1. So they have 3 big B's. And how many little b's? Well, the heterozygous individual had 1, and then the homozygous recessive individual had 2. So there's 3. So the frequency of the dominant allele in this case would be 3 over 6 total, which would be 1/2, or 0.5. And the frequency of the recessive allele would be, again, 1/2, or 0.5. Okay? Because in diploid organisms, you can be homozygous or heterozygous. It is said if all members in the population are homozygous for a given allele, so if every single individual is little b, in the whole population, or every individual is big B, that trait is said to be fixed because we can't actually pass down. If you're big B, and everyone is, there's no little b to pass down, and vice versa. The same thing's true for little b. If everyone's little b, there's no big B's to pass on, barring a random mutation that could occur that could create it. Okay? If that happens, it's said to be fixed in the population because that's the only allele that's available to be passed down. So this is kind of showing you a visual representation of the idea of a gene pool in wild boars. So you can see, like, all the individuals, what are the alleles that they possibly have? Let's throw them in. That's the gene pool for the population. And we get our frequency of dominant and recessive traits by taking the total and taking the part, how many big B's out of the total number of alleles are there, how many little b's out of the total number of alleles are there. Okay, so this is where it gets a little bit more complicated. When we look at the Hardy-Weinberg equation, this is actual math that we do to test to see if evolution is occurring within a population at a specific locus or trait. And this is actually derived from a Punnett square, this equation. If we look at the standard Punnett square, you have between 2 heterozygous individuals, we have a big T, little t, right, on each side. So that means each individual was heterozygous. And you get, in that case, 1 box that's big T, 2 boxes that are big T, and then 1 box that is little t. So the same thing in an individual, those are all the possibilities. But what if we extrapolate that to the whole entire population? Everyone does the same process of meiosis to create their gametes. So we can actually take the idea of a Punnett square, which we do for an individual, or 2 individuals that are mating with each other, and show the whole entire population. So if you start with a gene pool of possible alleles, and you assume that gametes form the same in all individuals through the meiotic process, you will end up getting in that population the same frequency as you would expect to see in a Punnett square. So the Hardy-Weinberg equation is actually p squared plus 2pq plus q squared equals 1, where p is the dominant allele frequency and q is the recessive allele frequency. So you may be like, well, how does that relate to that Punnett square we just did? So when you look at it, big T is actually called Q squared, or P squared, excuse me. It's P squared because P represents the dominant allele, and there's two of them, big T and big T. So that's the same as saying big T squared, or in this case, we're calling the dominant allele P squared. And there's only one box, so we don't put a subscript or a coefficient in front of it. Little t is really Q squared because Q represents the recessive allele. So there's two little t's, little t and little t, also known as t squared, or in this case, Q squared. But there are two boxes that are big T, little t. So we call it 2 times PQ, P being big T and Q being little t. So that's how we derive the Hardy-Weinberg equation. Now, together, the frequency of all of the alleles in a population is 1. That's like saying, like, if we take all of the dominant alleles and all the recessive alleles in a whole population together, when we add their frequencies together, it should be 100%, or 1 in terms of frequency, right? So, like if you have a P of 0.5, then Q has to be 0.5 because together P and Q have to equal 1. And this is only dealing, this specific version of the Hardy-Weinberg equation only deals with traits that have 2 alleles in a population. Once you get 3, it gets more complicated, and you don't have to know those types of questions really for the AP biology exam. Okay. Another thing that you can do to this, if you remember how to do factor quadratic equations, if you take this and actually do it, you get P plus Q squared equals 1. So if you factor that, that's what you get. And if you know that, if you take the square root of both sides, the square root of P plus Q squared ends up being P plus Q, and you take the square root of 1, that's 1. So P plus Q also equals 1. So this will be important to know that when we start to do some more problems with this in a little bit. All right, so what does this mean? So if a population follows Hardy-Weinberg equilibrium, that means that the frequency of traits is not going to change over the course of generations, or evolution is not occurring. So this is basically a test to see if we do not follow the Hardy-Weinberg equilibrium over the course of generations, we can test to see if evolution is occurring. So if you follow it, you're not evolving, or the population is not changing at that particular gene. But if it changes and it doesn't follow Hardy-Weinberg equilibrium, then evolution is not. Then evolution is occurring. So conditions that have to be the case if something is in Hardy-Weinberg equilibrium, meaning evolution is not occurring. The first thing, and none of these are actually met really in real life, but we like to assume that that's the case. So the first thing is no new mutations are springing up. We're not creating new recessive alleles. The next one is that mating is completely random between individuals. There's no mate choice. They're not selecting for specific traits. Okay. The other thing, no natural selection. There's no selection for a specific trait, increasing its frequency in the population. The population must be infinitely large, and we'll talk about why that is a little bit more when we get into genetic drift, because of random changes that can drastically affect populations. And then the other thing is that there's no gene flow between neighboring populations, which is called immigration or emigration. We don't have random alleles coming in from a neighboring population or leaving to go somewhere else. Okay? So you have to actually memorize all of these conditions. Rarely will they have to make you say all 5 of them, but you should have at least several of them on hand. And it's anything that could affect allele frequency over the course of time. All right, so now we're going to talk about how we solve these actual problems. So here's a question that would be something that they could give you based on Hardy-Weinberg. And this is actually a very simple one, and we'll get a little bit more complicated. So suppose that in a plant population that red flowers are dominant to white flowers, big R to little r. In a population of 500 individuals, 25% show the recessive phenotype. How many individuals would you expect to be homozygous dominant and heterozygous for the trait? All right, so the first thing you have to remember is this is a population that is not evolving, and we're going to assume that the conditions, all the conditions that we talked about on the previous slide are met. Okay, so we're going to say this is if evolution is not occurring, this is how many individuals in a population we would expect to have specific different genotypes and phenotypes. So the first thing, the way you solve every single one of these problems, pretty much, is we need to find Q. Or in this case, Q squared, which we can then find Q from. So remember, Q represents the recessive trait frequency. Q squared represents or the recessive allele frequency. Q squared represents the homozygous recessive genotype. And the reason we use Q squared is because you have to be, for example, little b, in order to show the recessive trait. If you're big B, or big B, you're going to show the dominant one. So we know what Q squared has to be based on looking at organisms. So all of the white flowers have to be Q squared. So if they say, in this case, 25% show the recessive phenotype, then Q squared must equal 0.25. How do we get Q by itself from that? We have to take the square root. So square root of Q squared, in this case the square root of 0.25, now Q must equal 0.5. Now once we have Q, we can find P fairly easily, because remember, when you factor the P squared plus 2PQ plus Q squared equals 1, when you factor that, you end up getting a P plus Q equals 1 situation. So you can now plug Q in, which is 0.5. So P plus 0.5 equals 1. So that means P must also equal 0.5. So the frequency of P is 0.5, the frequency of Q is 0.5. And now we can actually figure out our expected homozygous dominant and heterozygous phenotypes. So the frequency of the homozygous dominant would be big T, or big R, I should say. I know I wrote T's on here because I was going back to the box that we did initially. But it would be like big R, or P squared equals the frequency of the P allele times the P allele, which would be 0.5 times 0.5, which is 0.25. For the heterozygous individual, it's 2 times P times Q, so it's 2 times 0.5 times 0.5, which together equals 0.5. And we already know Q squared, which is 0.25. So collectively, when you plug all of that in, you end up getting a total of 1. 0.25 plus 0.5 plus 0.25 equals 1. And at the same time, your recessive allele, P, or Q, is equal to 0.5, and your dominant allele is also equal to 0.5. Together equal 1. That's how you can check your work to make sure you did this right. So how do we turn these frequencies into actual numbers in the population? Well, let's say there's 500 individuals in the population. Well, we'd expect 0.25 of them to show the homozygous dominant phenotype. So 0.25 times 500 is 125 individuals. How about for big T, little t? Well, we said that was a frequency of 0.5. So 2 times 0.25 times 0.25 times 500 total equals 250. So collectively, if you add up the number of homozygous recessive, of homozygous dominants, and heterozygous individuals, which are the only 3 conditions we can have if it is, if there's only 2 alleles here, you get a total of 500 individuals, back to the original total number of individuals we had at the beginning. So that one was easy. Let's try one that's much more difficult, okay? The numbers don't work out quite as well in this case. So, a rather large population of biology instructors have 396 red-haired individuals and 557 gray-haired individuals. Assume that red hair is totally recessive. Please calculate the following: the allele frequencies of each allele, the expected genotypic frequencies, the number of heterozygous individuals that you would predict to be in this population, the expected phenotypic frequencies, and then a last part of the question, which is conditions happen to be really good this year for breeding, and next year there are 1,245 young potential biology instructors. Assume that all of the Hardy-Weinberg conditions are met. How many of these would you expect to be red-haired, and how many would be gray-haired? Okay, so let's step you through each one. The first thing we're going to do is we're going to calculate the allele frequencies for each allele. Remember, P and Q are your alleles. So I have the information up top. Again, there are 396 red-haired and 557 gray-haired individuals. The first thing you always do for these problems, remember, is find Q squared, or the recessive trait. In this case, not the allele yet, the actual trait itself. In this case, 396 are red hair, and remember, red is recessive. So what's the frequency there? 396 over the total, which is 396 plus 557. And when you do that calculation, you get a Q squared frequency of 0.416 if we round to the nearest thousandths. Next, we need to find Q. So you take the square root of Q squared, and in this case you get a Q of 0.645. You next need to find P. Remember, P plus Q equals 1. So P plus 0.645 equals 1, and if you solve for P, you end up getting a P of 0.355. So P plus Q should equal 1 collectively. Those are your allele frequencies. Now that we found both P and Q, we can find the phenotypes and genotypes specifically of the homozygous dominant and the heterozygous individuals in the population. So next, we want each genotypic frequency, P squared, 2PQ, and Q squared. We already know Q squared is 0.416. We figured that out in the first step. But how about P squared? Well, we just calculated P, which is 0.355. We square it. You get P squared to be 0.126. 2PQ, you take 2 times 0.355, which is the frequency of P, times 0.0645, which is the frequency of Q, and that you end up getting a 0.458. And then you can check your work at this point. Now, because we rounded to the nearest thousandths and not the 10,000th place, it doesn't perfectly add up to 1, but it's very close. If you would have gone to the 10,000th place, there are four places after the decimal, you would have ended up getting exactly 1. Okay, but it's very close to 1. So that's how you know you're right, or you did the calculation right. So now we want to figure out the number of individuals of that population we would expect to be heterozygous. So in that case, you take the frequency of heterozygotes, which is 0.458, and multiply it by the total number of individuals in the population, which is 953, which we got from adding the 557 plus the 396 up top. And in that case, we'd expect 436.5 individuals to be heterozygous. But we can't tell. Remember, you wouldn't know if they're heterozygous or not. That's just what the expectation would be if all the characteristics of Hardy-Weinberg were followed, or all of the stipulations of Hardy-Weinberg were followed. And you can't have a half of an individual, too, so this is an estimation kind of thing. For the accepted phenotypic frequencies, all we have to do is we figure out the homozygous dominant and heterozygous together, which show the dominant phenotype, right? So because big T, and big T, little t all show the dominant trait. So we take our P squared plus our 2PQ frequencies, so 0.126 and 0.458, and you get 0.584, or this should be about 58.4% of the population should show the dominant phenotype. But we already knew that, though, because if you would take the original 557 of the gray-haired individuals, right, those are the ones that show the dominant trait, and you divide by the total number, you actually end up getting 0.584. This is just showing you how it kind of works out. So in the last step, it said conditions happen to be really good this year for breeding, and next year, remember, this is just completely ridiculous. It's just a fake problem, obviously. Next year, there are 1,245 young potential biology instructors. Assuming that all of the Hardy-Weinberg conditions are met, how many of these would you expect to be red-haired, and how many would you expect to be gray-haired? Well, in this case, if it's in Hardy-Weinberg equilibrium, that means from generation to generation, there is no change in frequency. So the frequencies of everything should stay the same. So all we have to do is take the total population, in this case, 1,245, and multiply it by our frequencies before. Okay, so the red-haired individuals is Q squared. So you take 0.416 and multiply by the total, which is 518. Then you take the gray-haired frequency, which is 0.584, which we calculated just previously, multiply that by 1245, and you get 727. And together, because those are the only two traits you can have, that should equal the total number of individuals in the population. Does it equal each other? Or does 1245 equal 1245? Yes. So you're all good. So this is something we're actually going to go over a little bit more in lab. But how do we know if evolution does occur? If Hardy-Weinberg is a measurement to show that if it stays within it, Hardy-Weinberg means that evolution does not occur, then how can we tell if evolution does? Well, we can use a chi-squared analysis, because you can say, okay, based on this generation, we'd expect to get these frequencies. Now, what if the next generation happens and we get completely different things from what we'd expect in Hardy-Weinberg? Well, you can do a chi-squared and do your observed minus expected over expected squared, or squared, this is not typed right, observed minus expected squared over expected. And then we get our observed frequencies. We compare them to our Hardy-Weinberg frequencies based on initial numbers of homozygote recessives. And if the frequencies of the alleles differ significantly from what we'd expect Hardy-Weinberg to be, then evolution has occurred. Okay, we're going to practice that more in lab. So what actually causes these different changes in allele frequency? Well, one could be natural selection. Okay, so that's when we select for a specific trait, it provides an advantage for that individual. The example I gave before was the white allele in wolves, right? So if you're little b, if you're a wolf, you have white fur, and let's say there's particularly harsh winters now for some reason, and as a result of that, white is now selected for. So that would provide a benefit. They reproduce more often, pass those traits on to their offspring, so that white trait increases in frequency. That's an example of natural selection. But one of the cool examples of the way evolution can occur that has nothing to do with natural selection is called genetic drift. Drift is one of the most important things to understand in terms of evolution. This is random changes in allele frequency. So random things happening completely by chance, not because they have a selective advantage. Okay, so an example of genetic drift. So a giant tree falls on and crushes 40 random plants. It has nothing to do with their selective ability. The 40 random plants die. The rest of the population, let's say maybe there was a rare allele within those 40 and it just gets destroyed for no reason. That would normally provide it a selective advantage. Or maybe maybe there were like 20 plants left and there were rare alleles within those plants and they don't provide a selective advantage, but now they've increased in frequency in the population because we just destroyed 40 that didn't have that trait. Okay? So it has nothing to do, genetic drift, has nothing to do with the ability of the plants to reproduce and survive and pass their genes on to the next generation. There's two major types of drift that we're going to talk about. One is the founder effect, and that's when a few individuals leave a current population and repopulate a completely different area. The Amish would be an example from that. A lot of the groups that left England and came to America would be examples of that specifically. But the interesting thing about the Amish population is that they specifically do not really breed with outside of their group. And because of that, they had a specific set of recessive traits that were more common in them than in the general population. So when they went and founded a new colony, the frequency of certain recessive traits was very high with them. One of them is polydactyly, which is actually an increased number of fingers and toes. So you actually see that within the Amish population. And that's an example of genetic drift. In this case, though, you can see this is just a random example. You have 20 pink and 10 red in the initial population. A small subset leaves. It doesn't matter, red or pink didn't matter. It's just a random subset left to go found a new population. But now we have way higher frequency of red compared to pink. And then when they reproduce, not due to any selective advantage, the red is going to be a higher frequency within that population, or the red allele will be. Another example of this is a bottleneck. It's very similar to the founder effect. The only difference is, in this case, instead of a small group going and founding a new population, some sort of natural disaster comes in and destroys almost everyone except for just a few individuals. You can see in this specific bottle, right, we have different color or different traits. We have white, we have blue, and we have yellow. Now, yellow is in that population, but it's a fairly rare allele, but it exists. But when they did the bottleneck and they killed off a majority of the individuals, notice what was left. There originally was about a 50-50 split of blue and white and a few yellows mixed in there. Now we're dominant blue over in the surviving population with only one white. This is going to drastically affect those traits in the population. And those traits likely had nothing to do with their ability to survive. This is like a freak kind of event that happened that caused this bottleneck. Another thing that can affect allele frequency, as we said, is immigration and emigration, or gene flow as it's called, between neighboring populations. What happens is if two populations are isolated from each other, then there's differences in their allele frequencies. They could eventually actually become a different species over the course of a long time if there is no gene flow between them. because they kind of go on different evolutionary paths. But if there is gene flow, it tends to reduce genetic diversity between two groups. It's like mixing and shuffling them over and over and over again, and it should keep the allele frequencies about equal or at least similar to each other in both populations. Okay. Something I want to drive home though. Natural selection is the only natural process that consistently will cause adaptive evolution though, into which it's not purposeful, but nature selects for those traits that are better at surviving and reproducing within that particular environment. Drift can cause change. Sometimes that's adaptive, but that would be a completely random chance if it was. It doesn't mean it's not important. Genetic drift is a very important mechanism for evolution, but it doesn't drive adaptive change. Okay. There are actually several different types of natural selection that occur. I'm going to try to explain different examples of each. So one is directional selection. That's where if you kind of have, these different traits, you're going to shift towards one extreme or another. Everyone's going to shift towards that way. So like human height would be an example of this. We are selecting for taller individuals overall over the past 200 years or so. And you select against individual with one extreme or the other. So another example of this, not even humans, taller trees can outcompete shorter trees. So taller trees are selected for because they can tend to get more light. And over time, you'd expect the population of trees to get taller and taller because the ones that were taller collected more light, were able to do more photosynthesis and create more glucose, have more sugar, create more babies, and thus they have a higher reproductive success. Another example would be disruptive selection. So we talked about directional, now it's disruptive. This is where the phenotypic average is selected against. So an example of this would be, let's say there's a bunch of insects that live in a rocky beach, okay? And I'm completely making this up. But there are really dark rocks on the beach, like black rocks, and then there's really light rocks on the beach. And there's 3 different color morphs of this insect. There is a black insect, there's a white insect, and then there's a gray insect. In this case, the black insects can blend in on the black rocks, and the white can blend in on the white, but the gray don't blend in on either. So gray would be selected against. They would be found more often by predators. And as a result of this, because gray is selected against, you end up getting the population kind of split into individuals that are black and then individuals that are white. And what's really cool about this is that you would actually end up probably creating a different species in this case, because they would have different niches, as they're called, because the black ones would only live on the rocks that match with them, so they weren't seen by predators, and then the lighter colored ones would only be on the lighter colored ones. And maybe that would cause them to stop mating with ones of that of the different color, just because they don't live on the same rocks anymore. So over time, that could be an example of something that causes a speciation event. The last type is called stabilizing selection. That's where we select against either phenotypic extreme. So in this case, it would be the opposite. They're only gray rocks. So if you were too light or too dark, you would be seen by predators. Another example would be birth weight in babies. A child that's too small would be too weak to thrive. A child that's too big wouldn't be able to get out. So we have this kind of narrow range in which babies at term would be in humans. So here's showing you all 3 types and how you're selecting for would be the increase in frequency of the allele, selecting against would be the decrease for the down arrow, and what would end up happening to the populations in each case. One of the examples of this actually is the peppered moth. This is a real life example. There's 2 colored there's 2 color morphs of the peppered moth. There's this really dark gray colored pepper moth, and there's this really light colored peppered moth that looks like it's peppered, basically. And these peppered moths actually live on trees in England. Now, they like to kind of live on the lichens, and you can see the lichen is this stuff that's right here. Notice how you can barely even see it, but there's that the lighter colored morph of the peppered moth blending in really well, but we can't really see this darker colored one. And when there is no lichen, you can definitely see the lighter colored one on the tree bark, but the darker colored one is not as visible. So the story goes, the way the peppered moth kind of works is that during the Industrial Revolution, because of all the pollution that was being thrown in the air, the lichens were actually getting stained to be a darker color. So the original color morph of the lighter one used to be selected against or selected for because of the lichens were lighter in color. As soon as that lichen became darker, predators started seeing the light ones more often, and the dark ones ended up being the ones that were favored in that case. So what happened was the frequency of the dark allele increased in the population, and the lighter colored allele decreased in the population. Then, more recently, when we've kind of cleaned up the air, the lichens have gone back to normal color. We've now seen an increase back to the color of the lighter color allele and a decrease in the frequency of the darker color allele. So if natural selection is working, how do we maintain all this variation within a population? You'd think the best traits would eventually fix themselves. Like if there's a trait that's definitely better than another one, that trait would always kind of, over the course of time, eventually it should sort out that trait would fix itself, meaning there would be no examples of that other trait left. There's a few issues with this. One is that the environment's constantly changing, so the best traits that exist now may not be the best later, as you could see with the peppered moth. There are some other things that could be going on as well, and one of them is that many organisms are diploid, right? So harmful alleles that are dangerous can be masked because you have a dominant and a recessive, and the recessive a lot of times is the harmful one. Not always, but a lot of times is. So if you're big A, little a, for example, you would show no phenotypic effect of having that harmful allele, but it can stay within you, and you can pass that on to the next generation. Another really cool thing is that some harmful alleles actually possess the ability to provide something called a heterozygote advantage. So I want you to think about what you think that could possibly mean for a second. And I'm going to tell you actually an example of it that exists today. So sickle cell anemia, horrible disease. If you have two little a's, for example, two recessive alleles, you will have serious problems and die very young from the disease within the first 5 to 10 years of your life if you aren't helped by medicine. So this, like in third world countries specifically. But also in Africa, malaria is a very serious problem. That is a parasite that actually can attack your red blood cells. Okay. But what's really cool is that they cannot attack the sickle cell red blood cells. So malaria actually does not have a very good effect, or doesn't have very much of an effect on someone who is heterozygous for sickle cell anemia. They are not a don't have full-blown sickle cell anemia, but they have one allele version of the sickle cell trait. So they have some cells that sickle, but a bunch of normal ones. And it's actually enough so that the malaria parasite can't thrive, and they become immune to the malaria parasite. So this actually causes stabilizing selection because we select against individuals that don't have the sickle cell allele because malaria could kill them. You select against the individuals that do have the sickle cell allele full-blown, little a, because they die of sickle cell anemia and never reach reproductive age. And you select for the heterozygous individuals. This actually shows you the frequency of the alleles throughout Africa. And you can see areas in which there are very, very high amounts of malaria, which would be in these areas specifically, but anywhere you see these this kind of dotted pattern is where malaria exists within Africa. And you can see the highest frequencies are right smack dab within this area in which malaria is an issue. So as I said, if you have a normal phenotype, big A, you'll die of malaria, or could die of malaria if you're infected. If you are little a, you have sickle cell disease and that'll kill you, but if you're heterozygous, it'll select for that because it gives you an advantage to survive. The last thing I want to talk about in terms of microevolution and evolution in general, there is no purpose to evolution. There's no, in terms of the way we think about it scientifically. We're not driving towards perfection. We're not driving towards anything. It's just differential survival and reproduction. What is beneficial today may not be beneficial tomorrow because of the constant changes in environmental conditions. Sometimes there's historical and time constraints to this, right? Like you have this one allele that looks like it's really good at surviving, and then for some reason a genetic drift occurs, destroys everyone with that allele, and then it's gone. It could come back again through random mutation, but the chance of that happening is somewhat slim. So just remember, there's no purpose to evolution. It's not a purposeful process. There's no, like, an organism doesn't purposely evolve, because organisms don't evolve, populations do. But populations don't purposely evolve. It's just because of a random mutation, which provides all of this variation, and selecting for individuals that have the mutations that are best able to fit and survive in that environment. All right, that's it.